In the name of Allah, the most beneficent and merciful. Here I am, Muhammad Khan, lecturer in botany, University of Education, Lahore. So today we will discuss a brief history and development of microbiology. So let's start. First of all, we discuss about the beginning of microbiology. Mosaic laws that found in the Bible about basic sanitation have been used through the centuries, and it still contribute in our practice for cleanliness, and also used as a preventive medicine. So the Bible also refers to leprosy and to the isolation of lepers. Although in those days the term leprosy probably included other infectious and non-infectious disease. Isolation did limit the spread of the infectious disease. So these Mosaic laws actually are the baseline for the beginning of microbiology. Here we have some history related to Greek. Greek also anticipated microbiology. A Greek physician, Hippocrates, set forth ethical standards for the practice of medicine that are still in use today. So he associated particular signs and symptoms with certain illnesses, and realized that disease couldn't be transmitted from one to another by clothing or other. The disease could be transmitted from one to another by clothing or other objects. Similarly, the Romans also contributed to microbiology as early as the 1st century. A scholar and writer, Varro, proposed that tiny invisible animals enter the body through the mouth and nose to cause disease. Similarly, another philosophical poet cited that seeds of disease in his book De Rerum Natura, on the nature of things. So early on, a bubonic plague, also called the Black Death, appeared in the Mediterranean region, where it reached epidemic proportion and killed millions of people. So the Black Death was a great leveler, and it killed rich and poor alike. Similarly, a disease of plague that affect most of the region of the world, especially Asia, Europe, Italy, and France, England, and finally Northern Europe. And it prevailed more than 300 years. The wealthy fled to isolate summer homes but carried plague-infected fleas with them in unwashed hair and clothing. Plague alone wiped out 25 million people, 1/4 of the population of Europe and neighboring regions, in just 5 years. Their bones are now displayed in an Ozore. So here we begin with the classical golden age of microbiology. A most prominent scientist in this age is Robert Hooke. One of the most important discoveries of biology occurred in 1665 with the help of crude microscope that is took place by Robert Hooke. So Robert Hooke stated that the life is life based on a smaller structural units that were cells. So Hooke's discovery marked the beginning of the cell theory that proposed the that all living things are composed of cell. On the basis of these discoveries or investigation, he would be able to investigate the structure and function of cell. The other prominent scientist of this age is Louie Van Hoek. He was a scientist that observed the microbes through a simple microscope. So his single lens magnified 15 to 300 time magnification. One of his achievements, he wrote series of papers describing his observation of bacteria, algae and protozoa and fungi through the help of his simple microscope. He found a small entity that he called as animalcules. He found them in stagnant water, in sick people, and even his own mouth. Over the year, Louie Van Hoek observed all the major kind of microorganisms. He once wrote, "For my part I judge from myself that all people living in our United Netherlands are not as many as the living animals that I carry in my own mouth this every day." So in 1670s, he wrote numerous letters to Royal Society in London and pursued his studies until his death in 1723 at the age of 91. Louie Van Hoek refused to sell his microscope to others and so failed to feed the development of microbiology as much as he could have. So after Louie Van Hoek death, microbiology did not advance for more than a century. Eventually, microscopes became more widely available and progress resumed. Now here we start the second golden age of microbiology, that based on era from 1857 to 1914. In this golden age, there is a creation of specific phenomena that is called as spontaneous generation. Early believed that some forms of life could arise from vital forces present in non-living or decomposing matter, abiogenesis. In other words, organism can arise from non-living matter. So they call this hypothetical process is known as spontaneous generation. But in 1688, Italian physician Redi performed an experiment. In this, Redi filled 2 jars with decaying meat. The first was left unsealed. The flies laid their eggs on the meat and the eggs developed into larvae. The second jar was sealed, and because the flies couldn't lay their eggs on the meat, no maggots appear. Still, Redi's antagonists were not convinced. They claimed that fresh air was needed for spontaneous generation. So Redi set up second experiment in which he covered a jar with fine net instead of sealing it. No larvae appeared. Maggots appeared only when flies were allowed to leave their eggs on meat. So in 1745, when John Needham, an Englishman, found that even after the heated nutrient fluid, chicken broth and corn broth, before pouring them into covered flask, the cooled solution were soon teeming with microorganisms. Needham claimed that microbes developed simultaneously from the fluid. 20 years later, Lazzaro Spallanzani, an Italian scientist, suggested that microorganisms from the air probably had entered Needham's solution after they were boiled. Spallanzani showed that nutrient fluids heated after being sealed in a flask didn't develop microbial growth. So Needham responded by claiming the vital force necessary for spontaneous generation had been destroyed by the heat and was kept out of the flask by the seals. Anton Laurent Lavoisier showed the importance of oxygen to life. Spallanzani observations were criticized on the grounds that there was not enough oxygen in the seed flask to support microbial life. After this, in 1858, when the German scientist Rudolf Virchow challenged the case for spontaneous generation with the concept of biogenesis, the claim that living cells can arise only from pre-existing living cells. Another scientist in 1961, Louie Pasteur, demonstrated that microorganisms are present in the air and can contaminate sterile solutions, but that air itself does not create microbes. And the other scientist, Louis Jablot, in 1670, conducted an experiment in which he divided hay infusion that had been boiled in two containers, a heated container that was closed to the air and a heated container that was freely open to the air. Only the open vessel developed microorganisms. This further helped to disapprove abiogenesis. Another two scientists also disapproved the process of abiogenesis. One is Schumann Friedrich Schroeder and von Dust. The other one is John Tyndall. And other scientist, that is Louis Pasteur, that play a vital role in the disproved of spontaneous generation of microbes by preventing dust particles from reaching the sterile broth. In 1861, he completed experiment that lays to the rest spontaneous generation. He also showed microbes called fermentation and spoilage. In Louis Pasteur's experiment, he filled several short-necked flasks with beef broth and then boiled their content. Some were then left open and allowed to cool in a few days. These flasks were found to be contaminated with microbes. The other flasks, sealed after boiling, were free of microorganisms. From these results, Pasteur reasoned that microbes in the air were the agent responsible for contaminating non-living matter such as broth in Needham flask. So, the content of the flask that possess S-shaped curve, in this flask, the broth in this flask didn't decay and showed no signs of life even after a month. So Pasteur showed that microorganisms can be present in non-living matter, on solids, in liquid, and in the air. He demonstrated that microorganisms can be destroyed by heat, and methods can be devised to prevent the microorganism to enter the nutrient environment. So that the technique that prevent contamination by unwanted microorganisms is to referred as aseptic technique, and that is a procedure that is adopted in laboratory and medical procedures. Furthermore, Pasteur found that microorganisms called yeast convert the sugar to alcohol in the absence of air. This process called fermentation that is used to make wine and beer. Souring and spoilage are caused by different microorganisms, and these microorganisms are called as bacteria. In the presence of air, bacteria change the alcohol in the beverage into vinegar in the presence of air. So Pasteur's solution to the spoilage problem to heat the beer and wine just enough to kill most of the bacteria that cause the spoilage. The process called pasteurization. So it is now commonly used to reduce spoilage and kill potentially harmful bacteria in milk as well as in some alcoholic drinks. This was the major step towards establishing the relationship between disease and microbes. So before the time of Pasteur, effective treatments for many diseases were discovered by trial and error, but the causes of the disease were unknown. So this is the main reason for the creation of germ theory of disease. The realization that yeast play a crucial role in fermentation. So this was the first link between the activity of microorganisms and the physical and chemical changes in organic material. The discovery altered scientists to the possibility that microorganisms might have some similar relationship with plants and animals, specifically that microorganisms might cause diseases. This idea was known as the germ theory of diseases. So the first proof that bacteria actually cause disease came from a German physician, Robert Koch, in 1876. He conducted an experiment, and that experiment is the base to formulate Koch postulates of disease transmission. Koch discovered rod-shaped bacteria, now known as Bacillus anthracis, in the blood of cattle that had died of anthrax. So he cultured the bacteria on nutrients and then injected samples of the culture into healthy animals. When these animals became sick and died, Koch isolated the bacteria in their blood and compared them with the bacteria originally isolated. He found that two sets of blood culture contained the same bacteria. Koch established a sequence of experimental steps for directly relating a specific microbe to a specific disease. These steps are known today as Koch postulates. So Koch postulates provide a framework for the study of etiology of any infectious disease. So the Koch postulates are: the microorganism must be found in abundance in all organisms suffering from the disease, but should not be found in healthy organisms. The second postulate is that the microorganism must be isolated from the disease organism and grown in a pure culture. The third postulate is that the cultured microorganism should cause disease when introduced into a healthy organism. And the fourth and the last Koch postulate is that the microorganism must be re-isolated from the inoculated disease experimental host and identified as being identical to the original specific causative agent. Now here we have the third golden age of microbiology, in which there is the invention of vaccination. So what is vaccination? Vaccination, injection of a killed microbe in order to stimulate the immune system against the microbe. And how vaccination came about. Edward Jenner in 1796 discovered that cowpox induced protection against human smallpox. This he called as procedure vaccination. So basically he conducted an experiment to find a way to protect people from smallpox, as mentioned in this slide. So inoculation of healthy individuals with weakened or attenuated forms of microorganisms that would otherwise cause disease to provide protection or active immunity from disease upon later exposure. So Pasteur and Rox reported that incubating cultures longer than normal in the lab resulted in attenuated bacteria that could no longer cause disease. So they working with chicken cholera. They noticed that animals injected with attenuated culture were resistant to the disease. After this, Chamber developed other vaccines, attenuated anthrax vaccine, in which he chemically and he provide chemical and heat treatment. Then he find out attenuated rabies vaccine, in which he propagated the virus in rabbit following injection of infected brain and spinal cord extract. So treatment with chemicals is referred as chemotherapy. Chemotherapeutic agents used to treat infectious disease can be synthetic drugs or might be antibiotics. So antibiotics are chemicals produced by bacteria and fungi that inhibit or kill other microbes. In 1928, Alexander Fleming discovered the first antibiotic. He observed that Penicillium fungus made an antibiotic penicillin that kills a fungi, that kills bacteria. In 1940s, penicillin was tested clinically and mass produced. So chemotherapeutic agents prepared from chemicals in the laboratory are called as synthetic drugs. A synthetic drug is also used for prevention from bacteria or microorganisms. Here we have the first synthetic drug. Paul Ehrlich speculated a magic bullet that can destroy the pathogens but not the infected host. He introduced an arsenic-containing chemical called salvarsan to treat syphilis. In the third era of golden history of microbiology, one of the most prominent progress is the discovery of antibiotics. So Alexander Fleming is a physician, Scottish physician and bacteriologist that produced antibiotic. Fortunately, he took a second look at the curious pattern of growth on the contaminated plates around the mold was a clear area where bacterial growth had been inhibited. Fleming was looking at the mold that could inhibit the growth of bacteria. This mold was later identified as Penicillium nodotum, later renamed Penicillium chrysogenum. So in 1928, Fleming named the mold active inhibitor penicillin. Thus, penicillin is an antibiotic produced by fungus. So here we have a photograph in this slide in which that clearly showed the action of penicillium as an antibody. Here these dots are showed. These are the bacterial colonies. But here this area are free from any bacterial colony. And here we have a penicillium colony. So it means that this penicillium restricted the growth of bacterial colony. So penicillium must have some type of antibodies or antibiotics that restricted the growth of bacterial colony. So in this slide, why drug resistance occurs? Drug resistance results from genetic changes in microbes that enables them to tolerate a certain amount of an antibiotic that would normally inhibit them. So these changes might include the production of microbes of chemicals, enzymes that inactivate antibiotics, or it may change in the surface of microbe that prevent an antibiotic from attaching to it, and prevention of antibiotic from entering the microbe. So here we have a brief description of different biologists that play a very important role in the development of different modern disciplines. So in the end, we have a very brief summary of all the history and development of microbiology from the era of 15th century to 19th century. So the different scientists play their role in the development of microbiology, and their contributions are highly appreciated till yet. And due to these scientists, we are able and capable to address different diseases and different phenomena that may be very harmful for us. That's all about for this lecture. May Allah protect us all from pandemic disease. Stay safe and healthy. Allah Hafiz.